is Emma Young, and I worked with Dr. Christine Anderson to test for the presence of tick-borne pathogens amongst mice and ticks at the Primmer Outdoor Learning Center, located in the Hocking Hills region of Logan, Ohio. Ticks are a leading vector in the United States, as they account for 75% of our vector-borne diseases. Lyme disease is the primary vector-borne disease in the United States, and approximately 300,000 people are infected by the disease annually. White-footed mice are the most efficient reservoirs for the bacterium Borrelia burgdorferi, which is a bacterium that causes Lyme disease. If mice are carrying the bacterium Borrelia burgdorferi, they are able to infect over half of the larval blacklegged tick population that feeds on them. A human can then contract Lyme disease when they are bitten by an infected blacklegged tick. White-footed mice are also reservoirs for Rickettsia rickettsii, which is a bacterium responsible for Rocky Mountain spotted fever. Rocky Mountain spotted fever is the most common rickettsial disease in the United States and is amongst the deadliest of the tick-borne illnesses in the United States. White-footed mice have been noted to greatly influence the presence of Rickettsia rickettsii in an area, and they are able to transmit the bacterium to the American dog ticks that feed on them. Humans primarily contract Rocky Mountain spotted fever when bitten by an infected American dog tick. However, other tick species can transmit the disease. Cases of Lyme disease and Rocky Mountain spotted fever are reported in the state of Ohio annually. The most common time of infection for these illnesses in Ohio is the summertime. However, people can contract these illnesses during any season. Lyme disease cases are more common in the eastern and southern parts of the state of Ohio, since that is where black-legged ticks are most commonly found. Rocky Mountain spotted fever can be contracted across most of Ohio, since American dog ticks are found throughout the state. The goal of this research was to analyze the presence of Borrelia burgdorferi and Rickettsia rickettsii among white-footed mice reservoirs and tick vectors at the Primmer Outdoor Learning Center. The data is important because it demonstrates the ecological dynamics between the pathogens, their vectors, and their reservoirs in southeastern Ohio. This data could also be compiled to create agent-based models that simulate the dynamics of these pathogens to determine the risk of transmission to humans. The Primmer Outdoor Learning Center was used as the field site for this research. A map of the property is shown in Figure 1. The property itself consists of 74 acres and has a representation of multiple ecosystems, including a temperate secondary growth deciduous forest, a fencerow habitat between a restored prairie and an adjacent agricultural field, a pine and spruce plantation, a riparian forest, 12 acres of restored prairie, multiple groundwater springs and streams, the Hocking River system, and a 15-acre wetland with a bald eagle nest. Multiple student-centered research projects have been conducted at the Primmer Outdoor Learning Center, and the property itself has also been used for educational and service-based activities. For the sample collection, 24 Sherman non-folding live capture traps were placed in the fencerow habitat, and 32 traps were placed in the woods habitat for a total of 6 live trapping sessions that occurred throughout June 2020 to July 2020. The traps were set and baited with oatmeal between 6 p.m. and 8 p.m. and were checked the following morning prior to noon. Any mice that were captured in the traps were identified and tagged with Monell ear tags in the field, as shown in Figure 2. A small tissue and blood sample were also taken from the tips of the mice's tails, and any excess bleeding from the tails was stopped by applying pressure and cornstarch to the tail tips before the mice were released. If any ticks were found on the mice, they were collected by removing them with a clean pair of tweezers and were stored in 70% ethanol. Ticks were also collected from the environment in 4 different sampling locations at the property with a dry cloth, which is a 1 meter squared piece of fabric attached to a dowel rod and rope. Dry cloth was drug across the ground for ticks to attach themselves to the cloth, as seen in Figure 3. All collected ticks were identified based on their species and life stage, as demonstrated in Figures 4 through 6, before they were stored in 70% ethanol. In the laboratory, DNA was extracted from all blood and tissue samples with the QIAGEN DNeasy blood and tissue kit. Polymerase chain reaction, also known as PCR, was used to determine the presence of Borrelia burgdorferi and Rickettsia amongst the samples. Each of the PCR products were then confirmed on 3% agarose gels that had been soaked in ethidium bromide and then imaged using a UV light box. A sample was positive for the presence of Borrelia burgdorferi if a 790 base pair fragment was present on the gel, and a sample was positive for the presence of Rickettsia if a 247 base pair fragment was present on the gel. For the results, a total of 10 different mice were caught and tagged in the fencerow habitat, and 8 different mice were caught and tagged in the woods habitat. None of the captured mice tested positive for carrying Borrelia burgdorferi. However, a female mouse from the woods habitat tested positive for Rickettsia, as shown in Figure 7. A total of 16 ticks have been collected over the course of the study as well. 10 of the 16 ticks were adults and have been collected from the sampling location in the fence row, on the mice in the woods, and the fence row, and on me. The remaining 6 ticks were nymphs and had been collected along the path within the woods, the path between the fence row and woods, the path near the woods, and from a mouse in the fence row. 14 of the 16 ticks collected were positive for Rickettsia, as shown in figures 8 and 9. This study sought to determine the presence of Borrelia burgdorferi and Rickettsia amongst mice reservoirs and tick vectors at the Primmer Outdoor Learning Center. White-footed mice have been recognized as efficient reservoirs for Borrelia burgdorferi and Rickettsia rickettsii. The populations of these mice have been extensively studied at the Primmer Outdoor Learning Center for 8 years. This is the first year since 2012 to have confirmed the role of white-footed mice as reservoirs for Rickettsia at the Primmer Outdoor Learning Center. The vector for Borrelia burgdorferi is the black-legged tick, and these ticks are commonly found in the eastern and southern areas of the state of Ohio. However, the results suggested there was not a high presence of black-legged ticks or Borrelia burgdorferi at the Primmer Outdoor Learning Center. American dog ticks also serve as a vector for Rickettsia and can be found throughout Ohio. Based on the results, there was a presence of American dog ticks and Rickettsia at the Primmer Outdoor Learning Center. The samples that were positive for Rickettsia will be sent for genetic sequencing to confirm which species of Rickettsia were present in the mouse and ticks. In addition, some samples will be run on a gel again via gel electrophoresis due to unclear band presence. Finally, all samples will be tested for the presence of Francisella tularensis, which is a bacterium responsible for tularemia. Finally, I am extremely grateful to the individuals that have provided support for this project. Capital University Summer Scholars Program, Capital University's Boyd Fund for Undergraduate Scholarship, and the Primmer Fund have all generously provided funding for this research. I am thankful for the funding provided, as the project would not have been able to be conducted without the funding. In addition, I am appreciative to Dr. Nathan Walker and Dr. Matthew Buckholtz for their expertise and for providing positive control samples. I am also thankful to Back to the Wild in Castalia, Ohio for providing a tick in order to test DNA extraction methods.